family, and to other active people at learning of intellectual exploration as collaborative research, where distinguished research is their insight and knowledge behind. But before this expenditure begins, in-house household proceedings and basic coverage should not begin, it will be good and noble for us collectively recite basmalah together. Bismillah. My village audience, before we come into the main session, let us study for the structure as follows. The first, opening. Second, singing Indonesian national anthem and continuously with bars of BPH. We'll come and preach the material education and at the last closing. So, ladies and gentlemen, as we continue our program, I kindly request all of you to rise for a moment of national pride as we sing together Indonesian national anthem, Indonesia Raya. Following this, we will proudly present the anthem of our beloved institution, Bakti Tunas Pusara. Kindly remain standing until both anthems have been sung, after which you may be seated. And now, without diminishing the respect of our audience, I kindly request all of you to stand up, please.
My beloved audience, without further ado, it is an honor to invite to the stage to the esteemed figure of Rector Bhakti Tunasusada University, the deferred leader of our state institution, to deliver the opening remarks and extend to heartfelt welcome to all present. To Mr. Dr. Ruswanto, M.Si, the stage is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillahirabbil alamin. Wassalatu wassalamu ala asyrofil mursalin wa ala alihi ajmain. Alhamdulillah ya, pertama-tama kita ucapkan syukur pada Allah ya yang pada hari ini masih melimpahkan nikmat ya, rahmat, sehat dan kepada kita semua sehingga pada hari ini bisa bertemu di acara kuliah tamu ya. Salawat dan salam semoga tetap tercurahkan kepada junjungan Nabi besar kita, Nabi besar Muhammad sallallahu alaihi wasallam. Yang terhormat Ibu Yayah selaku Ketua Yayasan Bacteriological Tasikmalaya yang alhamdulillah sudah bersedia mendampingi di acara kegiatan pagi ini. Yang terhormat juga Pak Dekan ya, Pak Dekan Fikas, kemudian Ibu Dekan Farmasi, kemudian Ibu-ibu Kaprodi S1 Farmasi dan S1 Analis ya. Kemudian yang terhormat juga Prof Didik ya yang sudah memberi kesempatan bagi UBTH ya untuk untuk menjalin silaturahmi dengan SIBA University. My Honorable Associate Professor Akiko Takaya. Ya. Alhamdulillah. Kegiatan hari ini mungkin salah satu kegiatan pembuka ya bagi UBTH ya. Untuk ke depan nantinya kita bisa membuka kesempatan yang lebih luas lagi ya. Makanya kami harapkan khususnya mungkin kalau di sini S1 Farmasi dan analis kesehatan ya harus bisa merencanakan ke depan nantinya mau ngapain nih ya. Dengan agenda hari ini harapan kita ke depan semoga dari S1 Farmasi ya, kemudian D3 Analis ya, silakan rencanakan dengan sebaik-baiknya untuk perencanaan biar ke depan nantinya kita bisa langsung mungkin kerjasama ya ataupun berikutnya mungkin implementasi ya ataupun kegiatan-kegiatan lainnya. Mungkin bisa kegiatannya untuk dosen ya ataupun untuk mahasiswa ya. Insya Allah kalau kita BTH ya sekarang sedang semangatnya mungkin kita sedang menjalin kerjasama ya baik di nasional maupun internasional ya. Makanya kita juga harus satu frekuensi ya. Kita di manajemen ya, kemudian di fakultas, di prodi ya, begitu juga sama Bapak Ibu dosen sekalian, begitu juga sama mahasiswa harus satu frekuensi. Jadi apa yang kita kejar, apa yang kita lakukan ya tadi harus ada efek ke mahasiswa ataupun ke dosen ataupun nantinya otomatis ke kinerja perguruan tinggi ya. Makanya harapan saya pribadi sebagai rektor ya semoga nantinya kegiatan ini akan ditindaklanjuti ya untuk implementasi MOU ya ataupun implementasi kerjasama mungkin ke depan bisalah. Kalau sekarang kan tentang spesialisnya tentang Staphylococcus mikrobiologi ya. Kalau beberapa dosen kita kan ada bidang mikrobiologi ya. Makanya harapan kita ya silakan mungkin dosen ya yang bidangnya mikrobiologi cobalah ya menggali mungkin ada ada kemungkinan kerjasama dengan ke Jepang. Terus terang mungkin kalau di Jepang kita baru satu kerjasama kayak yang kemarin ya. Mungkin kalau ke depan nantinya kita bisa tindak lanjuti ya, kita akan memperbaiki lagi ya. Kita juga mungkin sudah sempat diskusi dengan Bu Dewi ya. Silakan rencanakan Bu Dewi untuk tindak lanjutan setelah kegiatan ini. Semoga sih ada kegiatan-kegiatan yang memang sebagai follow up kegiatan hari ini. Harapan kami semoga kegiatan ini bisa memotivasi kita semua ya, memotivasi saya pribadi ya, kemudian Bapak Ibu dosen ya, kemudian dekan fakultas ya, kemudian mahasiswa terutama ya, karena terus terang kita sudah lama ya kadang-kadang kita pengen susah cari mahasiswa juga. Kita kadang-kadang masalah kita di situ. Kita pengen banyak mahasiswa yang dikirim, cuma tadi banyak juga ternyata agak susah kita mencari mahasiswa yang punya yang mau kesempatan. Makanya kita tadi ya, alhamdulillah sudah beberapa tahun ini kita berkomitmen ya, baik dari untuk dosen ataupun untuk mahasiswa, silakan rencanakan kegiatan kita ya. Mungkin juga bisa untuk nanti ya, dosen untuk tugas belajar ya. Insya Allah kita secara manajemen membuka kesempatan bagi Bapak Ibu dosen ya yang masih S2 ya, silakan cari peluang dan kesempatannya ya. Insya Allah ya, kita akan sangat mendukung bagi Bapak Ibu dosen yang mau tugas lanjut apalagi ke luar negeri ya. Itu cita-cita udah lama lah kita pengen dosen-dosen yang merencanakan kuliah di luar negeri ya. Dengan Bismillahirrahmanirrahim ya, acara kuliah tamu hari ini kita buka, semoga lancar dan membawa keberkahan dan kemanfaatan bagi kita semua. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you for Mr. Dr. Swanto M.Si for his inspiring words and welcoming speech. Now, my beloved audience, as we prepare to excavate into the heart of today's discourse, it is my pleasure to introduce the moderator in the main session. But before we proceed, allow me to briefly present the curriculum vitae for our esteemed moderator, who will then gracefully take over the proceedings during the presentation of the material and discussion. Fully name and degree, Dr. Dewi Peti Virgianti M.Si. Career as a lecturer in Analyzed Health, diploma in BTH University, and then as a screener of cancer cytology cervix in female cancer program until 2008, and academic background, doctoral degree completion in 2022 from Biology ITB, Bandung, and then a research student in 2011 at Pukyong National University, South Korea. Master degree completion in 2013 from Biology, Unsud Purwakarta. Bachelor degree completion in 2003 in Biology, Unpad, Bandung. And then the publication, first penazine and quinolone are main compounds produced by Pseudomonas aeruginosa, and one during inhibitory interaction with Rhizopus microsporus, Unraised Journal 2023. And then Serratia rubidia as contaminant in laboratory environment, Nusantara Bioscience Journal 2021. And then new record of Stenotrophomonas as endosbion bacteria and Rhizopus, Universitas Journal of Biology Diversity 2020. Clitoria ternatialin extract as a natural pH indicator of mannitol salt agar medium. In second BTH Health Science International Conference 2020. And then last short communication, genetic diversity of the IGF2 gene as a source of genetic marker for halal authentication, Nusantara Bioscience 2011. So, this is time for Dr. Dewi Peti Virgianti M.Si to join in the stage, and I invite you to chart the event from this point forward. Thank you.
Thank you. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Okay, respectable, the chairman of foundation, Bakti Tunas Husada, Ibu Yaya, yes, and respectable director of Universitas of Bakti Tunas Husada, Pak Ruswanto, the honorable dean and the lecturer of pharmacy faculty and health science faculty, and honorable and beloved all students as participant in this international expert lecture class. Yes. So, ladies and gentlemen, my name is Dewi Peti. As moderator of today's international expert lecture class, I'm very pleased to see you. And here, today's class is about antibacterial drug development strategy based on bacteriological study. Okay, our speaker today is Associate Professor Akiko Takaya. She is a lecturer in Department of Infection Control Science, Graduate School of Pharmaceutical Science, Chiba University, Japan. Maybe Here I would like to read her curriculum vitae. Okay. Academic background, bachelor degree, completion in 1999 from Division of Microbiology, Faculty of Pharmaceutical Science at Chiba University. Master degree, completion in 2001 from Division of Microbiology, Faculty of Pharmaceutical Science at Chiba University. Doctoral degree, completion in 2005 in Pharmaceutical Science at Chiba University. And professional appointment or career from 2005 to 2009 as lecturer at Graduate School of Pharmaceutical Science, Chiba University. From 2009 to present as Associate Professor at Graduate School and Pharmaceutical Science, Chiba University. From 2020 to present as Associate Professor at Mycological Medical Research Center, Chiba University. And from 2023 to present, organizing Laboratory of Infection Control Science. And about license, Prof. Takaya has Japan National Pharmacy License. Okay, and about membership in professional and social and society is professional society. Prof. Takaya is a member of Japanese Society for Bacteriology and a member of Pharmaceutical Society of Japan. Maybe we can mention several lists of her publication. First, from in 2023, Prof. Takaya has publication. The title of publication is High Efficiency Single Cell Containment Microdevice based on fluid control in Micromachine Basal Journal. In 2022, Prof. Takaya has two publications in Organic Letter Journal and Phytochemistry Letter Journal. The title are Two Bioactive Compound, Uniformides A and B isolated from culture of Nocardia uniformis in the presence of animal cell, and New Polyoxygenate Cyclohexanase isolated from Euphorirufa and Sinamtanin B1 isolated from Nephelium hypholicum with trial-resistant overcoming activity. And in 2021, one more, and earlier, yes, before, Prof. Takaya has more publication that we can read in the information display here, okay, in the screen. So many publications. Okay, before the expert lecture class begins, let me inform how this class will be going. Yes. First, Prof. Takaya will be invited to present the material presentation. After that, there will be a question and answer session. So please raise your hand and state your name to give the question to Prof. Takaya. So now allow me to welcome our expert lecturer, Prof. Takaya, to deliver her presentation. Okay, Prof. Takaya, the time is yours.
Thank you. Hi. Selamat pagi. Yeah, that is Ohayo gozaimasu in Japanese. So good morning, everyone. So thank you for inviting me this very good university. I'm very surprised that so you have the different culture from that our culture. So I'm very fun during the Indonesia. So can I sit down during my lecture? Okay. Okay, so first of all, I'd like to thank Dean and the member of the university for inviting me and giving me the opportunity to present our study. So I'm talking about this title today. So, but before beginning my talk, let me introduce myself a little bit. Okay, I am Akiko Takaya from Chiba University in Japan. So as you know, Indonesia and Japan are very far from here. So it's approximately 5,500 kilometers. So, but it takes about 8 hours by plane. But I think it's too long because yesterday, I came here from Jakarta. So this is very, very long time from Jakarta. Okay. Now, so but the weather is very different from here to Japan. So Chiba is now winter season. So last weekend it snowed. Okay, this is the snow. Oh, it is very cold now. So this weather is like September in Japan. It's not hot for me. Okay, the shape looks like so here is Chiba. So Tokyo is here, and so the shape looks like a bear. So Chiba's character is this bear called Chiba-kun. But the most famous bear character in Japan is Kumamon. So maybe someone knows. Okay, so I said Chiba is next to Tokyo. So there are some facilities named with Tokyo, even though they are located in Chiba. For example, the Tokyo International Airport is located in Chiba, and Tokyo Disneyland are located in Chiba also. Okay, and surrounded by ocean, it was a surfing venue for the Tokyo Olympics. And the Chiba University was established in Chiba and 75 years ago. So our university has 4 campuses. The main campus is Nishichiba, and there are also Kashiwanoha and Matsudo. Now I am researching at the Graduate School of Pharmaceutical Sciences and the Mycological Medical Research Center on the Inohana campus. So our Faculty of Pharmaceutical Sciences has a longer history than the university, having been established 130 years ago. Okay. So in my graduate school, we have about 20 laboratories divided into several research organizations. And so until last March, I belonged to the Laboratory of Natural Products Chemistry. But since last April, I have been learning the Laboratory of Infection Control Science. Okay? So now I'm working with these members. So a member is a student, PhD student, and the master degree student, and bachelor course student. So the aim of our study is here. So in line with our aims, we are currently researching these topics. So one of my research topics is about salmonella. So, but it's not a familiar in Japan, salmonella infection. But you have many salmonella infections, disease. So next time I want to talk about the salmonella infections. Okay, so among them, today I would like to about the research on crumb sensing in Staphylococcus aureus. So especially I will describe the 2 topics. The first topic is about research on the relationship between crumb sensing and the disease development. The second one is the crumb sensing inhibitor, which we have recently identified. Okay? Okay, I start. So I will show you the brief introduction about the bacterial crumb sensing system. Crumb sensing is a gene regulation system which senses the density of bacteria of the same species as itself and controls the production of various factors depending on the density. A molecular sensing density is called an autoinducer. The bacteria produce and secrete autoinducers to the outside of the cell. When the amount of autoinducer is low, in this case, that is when the number of bacteria is low, it suppresses the expression of various factors, thereby avoiding detection of host immune systems. So after bacterial growth, when the amount of autoinducers exceeds a certain threshold, that is when the number of bacteria has grown sufficiently, bacteria drive to excess virulence factors. So this system contributes to pathogenesis by expressing various factors at once. Okay. The chromosomes of Staphylococcus aureus transcriptional units control both AGR, B, D, C, A. This expression responds to an autoinducer peptide, AIP. The P3 activation is also dependent on AIP. It controls transcription of the regulatory RNA effector, RNA3, which drives the expression of many virulence factors. A propeptide translated from AGRD, since AIP is transported by AGRB and acts as a signal molecule. AGRC and AGRA constitute the two-component signal transduction system. AGRC is autophosphorylated upon the binding of AIP. The amount of AIP increases with bacterial density of the response regulator of AGRA by AGRC. Then AGR. Okay. So many studies about Staphylococcus aureus have shown that AGR chrome sensing is involved in bacterial virulence. However, little is unclear whether AGR chrome sensing function is directly related to disease development. So from our two studies, we found that AGR chrome sensing function is essential for disease development. One is a study on atopic dermatitis, and the other is a study on nosocomial infection. First, I will talk about a cohort study related to the development of atopic dermatitis called AD. Okay. The epidermis of skin plays a critical role in maintaining the barrier function and protecting the body against invasion by harmful microbes. When it is broken, Staphylococcus aureus can colonize and then cause AD. Several studies about Staphylococcus aureus have them focusing on the specific virulence effect for inducing AD. However, while Staphylococcus aureus selectively colonized in AD skin, but not in normal human skin, remains totally unknown. To investigate this question, we did an infant cohort study. In Japan, there is a medical checkup at 1 month, 6 months, and 1 year old. At that time, we counted the bacterial number on their checks and kept all of them. Then we examined the frequency when infants were diagnosed with AD at 1 year old. So here I show the frequency of bacterial colonization in 1 month and 6 month infants and AD development at 1 year old. Staphylococcus aureus in 1 month did not associate with AD development. In contrast, Staphylococcus aureus colonized 6-month infants had significantly higher odds ratio for AD compared to bacteria-negative infant. This result indicates that Staphylococcus aureus colonization on skin has already been regulated between 1 month and 6 months of age and associated with AD development. Okay, to understand the genetic factors of Staphylococcus aureus that are important for skin colonization, we performed whole genome sequencing of these strains. We totally analyzed the genomes of 242 strains isolated from infant skin at 1 month and 6 months of age before disease onset. So this slide shows you the rooted neighbor joining tree using a single nucleotide polymorphism called SNPs in the core genome of the strains. Okay. Red indicates strains from AD infants, and the black indicates strains from non-AD infants. The results suggested that phylogenetic diversity of the strains that colonize the skin is not different between AD or non-AD. To assess the acquisition of mutations in the genome during skin colonization, we identified the strains that were continuous colonizers. That means the same clone was obtained from the same infant at 1 and 6 months of age. And we identified 24 non-AD infants and 9 AD infants having continuous colonizers clones. In the AD infant, continuous colonizer detected 9 clones from 17 cases. In the non-AD infants, continuous clones detected 24 clones from 86 cases. Thus, a percentage of AD colonizer is higher than the of non-AD colonizer. Okay. To investigate the evolution of the genome during skin colonization, we analyzed the SNPs in the full genome of continuous colonizers. So this result reveals that the AGR region was associated with differential acquisition of mutations in AD infants or non-AD infants. In non-AD colonizers at 6 months, there are more mutations acquired in the AGR locus than in other genome regions. 2 clones acquired from cyst mutations is in AGR-C, and 2 other clones acquired mutations with RNS3 and upstream of AGR-B. Furthermore, RNS3 expression was decreased in these clones compared to the 1-month clone. Okay, to further investigate the role of AGR mutations in the AD development, we next assess the presence of mutations in AGR locus. So this slide shows you the frequency of SNPs and insertion of deletions in AGR locus of all isolates belonging to AGR type 1 compared in the type strain. You can see that there was a higher number of the mutation in AGR locus of 6-month range, that is the blue, from non-AD infants compared to those of 6-month range from AD infants. To confirm the importance of AGR function in skin colonization, we performed Staphylococcus aureus skin infection mouse model. So this in vitro growth curve of wild type and AGR deficient strains were shown here. You can see that both strains grow similarly. However, 7 days after skin inoculation, bacterial number of AGR deficient strain was significantly decreased
compared to its wild-type strain. Furthermore, inflammatory skin disease of AGR-deficient colonization mice was dramatically diminished compared to the wild-type colonized mice. Therefore, these results clearly suggest that AGR function of Staphylococcus aureus is essential for the adaptation to the surface of the skin. Okay, in summary of our infant cohort study, in healthy human skin, loss of function mutation is induced in the AGR locus, which eliminates Staphylococcus aureus from the skin. That is, retention of AGR column-sensing function on the skin is associated with skin colonization and development of atomic dermatitis. Okay, next, I will talk about research on nosocomial infections. So, among the infections caused by multi-drug-resistant bacteria, infections caused by methicillin-resistant Staphylococcus aureus, called MRSA, are highly important to prevent in Japan and other countries. So, MRSA infections in hospitals can be a problem due to repeated outbreaks caused by strains with the same genotype. However, it is still unclear why these strains continue to survive and what properties they acquire during their survival. To reveal this question, we analyzed identical clonal strains detected for nearly 2 years in hospital neonatal intensive care unit NICU outbreak. Okay. So infants in NICU were screened for MRSA carriage once a week. The gray bar shows the patient's length of stay in NICU, and the orange means that the MRSA was detected during screening. And the blue indicates that MRSA was detected by culture test when the patient developed MRSA infection. Okay. Over 2 years, 35 patients were colonized or infected with MRSA. We divided the period into the 3 groups and analyzed 21 strains. As a result of 4 genome analysis, 18 strains belong to the same clonal complex, CC1 with SCC-MEC4, was a strain responsible for the outbreak, so orange is here. So we examined genetic mutations in these strains. We determined the complete genome sequence of CN in the earliest stage and investigated SNPs in the core genome of all strains. There were no common SNP mutations. However, we found that SNP mutations in 2 strains, CN8 and CN31, caused loss of function in the AGR locus. So we examined their AGR expression. When we examined AGR expression by culturing it under high aeration condition, this one, we found that only 2 AGR mutant strains had no expression. So blue strain had not expression of AGR. However, when the culture conditions were changed to low aeration, in addition of blue AGR mutants, orange AGR in the orange ADR orange strains, AGR was not expressed in the strange isolated later so there were no mutations in the AGR region of orange strains. So this result suggested that strains that continue to survive in the hospital change AGR expression in an environment-dependent manner. without mutation in AGR locus. So we named these strains the environmentally adapted AGR called EAAGR. And we further analyzed its properties. Okay. So some previous studies have been suggested that decreased AGR expression may contribute to evasion from high concentration of antibiotics. Therefore, we examined survival in the presence of a high concentration of vancomycin. There were more viable bacteria in the AGR mutant strain than in the AGR positive strain. AGR strain, positive strain is red, and the blue one is AGR mutant. There were more viable bacteria. And the appearance rate of EAAGR strain is orange, was similar to that of the AGR mutant strain. So this suggests that the EAAGR strain can survive under high concentrations of antibiotics. Okay. We further examined the possibility that the properties of EAAGR contribute to its evolution in the hospital. Even if bacteria is the same species, different clonal complexes have different restriction enzymes and methyltransferases. So it is difficult to transform plasmid DNA extracted from the clonal complex 8 strain into the AGR positive strain of clonal complex 1. So the result is red bar. However, in the AGR mutant strain and the EAAGR strain, the number of transformers significantly increased compared to the AGR positive strain, even with plasmid extracted from the clonal complex 8 strain. In the clinical isolates, we found that the same resistant gene in the clonal complex 8 strain, CN12, was found in the later EAAGR strain, CN33 of C1 strain. So this suggests that EAAGR strain may take in foreign DNA and cause major genetic mutations, lowering its long-term survival. Okay, this is a phylogenetic tree shown again. The strain that evolved from CN2 continues to survive in the hospital environment by becoming an EAAGR. However, the AGR mutant strain is unable to survive despite showing similar properties of EAAGR strain. This suggests that EAAGR's expression may be necessary to survive within the host. So to investigate this possibility, we infected mice interperitorially with these strains. Half of the mice with the AGR positive strain died 7 days after inoculation. But most of the mice with the AGR mutant strain and with EAAGR strain survived. When we examined the number of bacteria in spring, 7 days after inoculation, almost little bacteria were detected in spring of mouse with AGR mutant strain. But many bacteria survived in that with the EAAGR strain. This result suggests that the EAAGR contributes to colonization and long survival within the host. Okay, this is a summary of 2 studies. In the skin, there are several commensal Staphylococcus species exist on the skin. Staphylococcus aureus AGR crumb sensing is necessary to compete with other commensal bacteria. Then, in atopic dermatitis, overexpression of AGR in Staphylococcus aureus produces toxins and then accelerate Th2 and Th17 type skin inflammation. In contrast, in the hospital and systemic infection, there are few competitors exist, and the silencing of AGR is a great advantage to survive against antibiotic treatment in Japan. However, AGR positive expression in Staphylococcus aureus is reportedly important for biofilm formation and escape from phagocytosis. And this is why EA-MRSA, but not AGR deficient mutants, survive in our systemic mouse infection model. Okay, from 2 studies, we demonstrated that AGR chrome sensing function is directly linked to disease in development. So compounds that inhibit AGR chrome sensing may be used for Staphylococcus aureus related diseases. Therefore, we screened compounds with AGR chrome sensing inhibitory activity. So at the beginning, I told you that our faculty had about 20 laboratories. There are several laboratories that specialize in organic chemistry. From these laboratories, we were able to collect approximately 20 compounds into our unique compound library. This is a result of comparing the structure characteristic of our compounds to FDA-approved compounds. So each molecule structure was analyzed using 792 descriptors and expanded in 2 dimensions using t-SNE. So blue one indicates our compounds, and gray one indicates FDA-approved drugs. So our compounds are scattered in features where there aren't many FDA-approved drugs. Okay, using these unique compounds, we screened compounds with AGR quorum sensing inhibitory activity using a reporter strain. The reporter strain has a luciferin producing enzyme driven by the P3 promoter in MRSA. So this shows the result of a screening of first 550 compounds. There were 85 compounds that did not activate P3 even after 24 hours of culture. When bacteria do not grow sufficiently, AGR is not activated. So therefore, we identified compounds that do not inhibit bacterial growth as compounds with AGR inhibitory activity. The results suggest that the 4 phytochemicals inhibit AGR expression. Among them, we investigated the molecular mechanism of AGR chrome sensing inhibition by physarin H. Okay, physarin H belongs to the class of physarins that exhibit promising pharmacology properties. We purified it from this plant. First, to assess the specificity of AGR chrome sensing inhibition by physarin H, P3 activity was examined in the presence of a serial concentration of physarin H, black S here. You can see that physarin H inhibits P3 activity in a concentration-dependent manner. In contrast, you can see that even 100 micromolar physarin H does not inhibit bacterial growth. This inhibitory effect was not affected even when excess AIP was added, shown red line. So next, we examined whether inhibition of P3 activity by physarin H affects the production of virulence factors. Now, we checked the hemolytic toxic activity. By real-time quantitative PCR, physarin H decreased the expression of RNA3 and AGRA, and the hemolytic toxins HLA and PSM alpha were also decreased. So we further examined hemolytic activity in the culture supernatant with Pfizerin H. The results indicated that Pfizerin H suppressed toxin production. These results strongly suggest that the reduction in AGR expression caused by Pfizerin H may reduce the prevalence of Staphylococcus aureus. So next we examined its target. As shown before, the inhibitory activity of Pfizerin H did not change even when AIP was accessed. This result suggested that the target of Pfizerin H was not detection of AIP, but its downstream. Okay, there are 4 different types of AGR in Staphylococcus aureus, but Pfizerin H inhibited the activity of all AGR types. Okay. The AGRA, the transcription factor, is conserved among the 4 types. So we thought the Pfizerin H may target AGRA. Okay. AGRA binds to the P3 promoter and activates its transcription. Therefore, we examine the effect of Pfizerin H on the interaction between DNA and AGRA using electrophoresis mobility shift assay. This is the result. Okay. When the mixture of DNA and AGRA was loaded, you can see that the complex was found. However, when Pfizerin H was added in the mixture, the interaction between AGRA and the DNA was not observed. You can see here. We further performed in-silico docking simulation to examine the interaction between Pfizerin H and AGRA. And histidine 169, aspartic acid 201, and arginine 233 of AGRA are important for DNA binding. From our simulation, it was speculated that Pfizerin H interact with N201 and adjacent R198 through hydrogen bonding. These results suggested that Pfizerin H binds to AGRN and then inhibits its interaction between AGRA and P3 promoter. Okay, to date, more than 70 Pfizerins have been identified. Some of them have pharmacological activity and others do not. So this difference in activity is not well understood. The oxygen atoms predicted the interaction between Pfizerin H and AGRA have a common structure to many Pfizerins. So thereby, we examined the AGR column sensing inhibitor activity of several Pfizerins. So in addition to Pfizerin H, 8 other Pfizerins were extracted. So blue indicates structural differences from Pfizerin H. When we examine these activities, Pfizerin 2, 3, and 4 inhibited AGR expression, but the others did not. However, all Pfizerins inhibited AGRA binding to DNA. So we examined their interaction with AGRA. As a result, Pfizerin H, 2, 3, and 4, which inhibited AGR column sensor activity, were predicted to have the same interaction. However, for the 5 Pfizerins that did not inhibit activity, the interaction position had changed. This suggests that slight change in the structure of Pfizerins changes the position where it interacts with AGRA, thereby changing its AGR quorum sensing inhibitor activity. Okay, finally, to investigate the possibility that differences in the interaction contribute to stability changes, we performed 100 nanosecond molecular dynamic simulations. Here I show you the result of Pfizer H with AGR quorum sensing inhibition activity and Pfizer 5 without AGR quorum sensing inhibition activity. Okay, RMSD result suggested that Pfizer H does not leave AGRA in the enduring simulation, but Pfizer 5 leaves AGRA in about 20 nanoseconds. So according to the result of RMSF and Pfizer 5, however, Pfizer H had more long bonding amino acid residues compared with the Pfizer 5. So this slide shows the interaction between AGRA and Pfizer H and Pfizer 5 every 20 nanoseconds. In the interaction with the Pfizer H, the interaction between the carbon oxygen at carbon 15 and losing 186 of GRA remained during this simulation. It was suggested that this interaction acts as anchor to a stable interactor with the DNA binding site. In contrast, in the case of Pfizer H, lysine 186 interacts with the carbon oxygen at carbon 26. It was suggested that this interaction does not remain in the DNA binding region, but rather pushes it outside from here. So we believe that elucidating the structure required for activity using similar compounds will provide important information when creating developing AGR quorum sensing inhibitors. Okay, I'd like to summarize today's talk. From 2 studies, we demonstrated that AGR quorum sensing function is involved in disease development. Furthermore, we identified several Pfizerins as an AGR quorum sensing inhibitor compound. However, our former studies suggested that Staphylococcus aureus avoids various stresses through reversible AGR quorum sensing expression. So complete inhibiting AGR quorum sensing can suppress the onset of this disease. But if AGR quorum sensing activity cannot be completely suppressed, bacteria may be able to escape stresses such as drug treatment. So therefore, AGR quorum sensing inhibitory compound may be candidate for the treatment of bacterial infections, but further research is required to realize this. Okay, so this slide shows our collaborators, and we have the many funding from these our government and these other foundations. Okay, I'd like to thank you for all. That's all. Thank you for your attention. Terima kasih.
Okay, thank you very much, Prof. Takaya, for giving us such informative and interesting presentation. And ladies and gentlemen, this is time to do and ask some question. And Prof. Takaya will answer one by one question, or we collect? Oh, directly. Okay. Okay, raise your hand, please, to ask the question. Oke, Pak Saiful, silakan.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good morning, Mrs. Eka Tanaka. Ya?
Yeah,
Okay.
okay.
Thank you for your presentation. I have some question about method and tool use the detection of analysis of EGR, accessories gain response. Okay. One question: what are the implication and application of your EGR identification and classification for the epidemiology, diagnosis, and treatment of Staphylococcus aureus? Second, how did the EGR group correlated and phenotypic characteristic and antibiotic resistance of your strain? Number three, what are the limitation, the challenges of your EGR identification and classification? And the last, what the future direction and recommendation for your research of the EGR Staphylococcus aureus? Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Sorry, this is the one more question. One by one. Okay, so first question is why?
Why application the tools of you use the EGR strain atau EGR in your strain?
Yeah, that EGR why we focus on the EGR chrome sensing target for the application of the treatment. Is it correct? Okay. So maybe some several study has focused on the EGR chrome sensing is the focus. So several studies has focused on the EGR chrome sensing for the target of the atomic dermatitis, because microbiome, unique microbiomes on the similar. So Staphylococcus epidermidis, AIP secreted Staphylococcus epidermidis prevent EGR chrome sensing in Staphylococcus aureus. And then Staphylococcus epidermidis avoid attachment of Staphylococcus aureus. So EGR chrome sensing cyst, if Staphylococcus if EGR chrome sensing of Staphylococcus aureus prevented using the chemical think the ADA development.
How about resistance of your gain in Staphylococcus aureus? Resistance of antibiotic?
No, that's antibiotic. So we usually we don't use the antibiotics for the atomic skins. So we usually use steroid on the other applications. So, but many cases in the atomic disease skin, so we can check, we can detect it the Staphylococcus aureus, but we can use it. And the second one.
Okay, thank you.
It's okay.
It's enough?
Enough.
Sudah, yang ke satu, kedua, nggak, udah terangkum.
Nanti lewat email aja lah. Supaya.
Via email. Okay, next question. Ya, Bu Tresna.
Okay, thank you for the nice presentation. That was a huge work, I think, from the isolation until the molecular mechanism, and then the in silico work that you did. But I'm curious, you said that in your presentation that you identified what I curious is that from activity?
Why? So you mean the why we another?
I mean, you work with various Visalin H, Visalin 1, 2, and others.
So
And the activity, I think, is different.
yeah.
Yeah.
I know. So this is the Visalin B, and
Yeah.
so, yeah, okay. So first we checked the Visalin H. So because so Visalin H, only Visalin H was in my fridge, so we can get the compound and this Visalin H put in our library. So we can check, we found that the Visalin H, first Visalin H has activity of AGR chrome sensing activity. So second, we extracted from same plant another Visalins. Okay, so now we got the Visalin B, so we can check the Visalin B and the Visalin H is the same activity against AGR chrome sensing inhibition, but Visalin B is antibacterial effect, so it is not good for the treatment for in the host with the mass AGR Visalin H and Visalin B. So, but we don't have any Visalins, so Visalin A or Visalin D. So we have not yet. We have not studied yet.
Oh, thank you very much.
Cukup, Putras? Yeah. Next question. Okay, last question. Okay.
So, hello, Professor. I wanna asking about your presentation. Is what are the most promising investigational therapies targeting quorum sensing in Staphylococcus aureus infections?
Let's repeat.
All right. What are the most promising investigational therapies targeting quorum sensing in Staphylococcus infection?
Promising investigation
Yeah.
on quorum
Investigational
sensing.
therapies.
The most promising
Sorry.
best, maybe the best. Bahasa Indonesia aja gimana?
Jadi terapi investigasi apa yang paling menjanjikan yang menargetkan pengideraan quorum pada infeksi Staphylococcus?
Jadi apa sih
Targetnya.
terapi untuk terapi ya?
Ya,
Therapeutic, the best therapeutic based on quorum sensing
Yes.
target in Staphylococcus aureus. Therapeutic berarti
Therapies,
aplikasi ya?
therapies,
Application.
Yeah, what the most
The most
promising
therapeutic strategy
for targeting quorum sensing?
quorum
Candidate of the target because AGR. So AGR is unique transcription regulator among the bacterial transcription regulator. So many transcriptional regulators like an AGRA has not common in all bacteria. So we think we hope the AGRAA is one of the best target of the AGR against AGR quorum sensing inhibition.
Okay. Is the AGR quorum sensing only in Staphylococcus aureus?
Yes. Yes.
Okay. Oh, so the best promising is there a promising compound, another